Hello everyone, I hope you're having a great time at the conference. My name is Davy Ouedraogo and I'm currently a PhD student at the University of Chicago in the Cobius Laboratory. So thank you for giving me the opportunity to present my research at the GLBIO conference. It's my pleasure to share with you TranscriptDB, a transcription database developed in our lab to explore and study the transcript conservation and their evolution within gene trees. So the interest of autologous genes has led to develop several genomic databases. So one thing in common is that they centered on genes. Also, they provide data and methods on gene evolution. In addition, we can retrieve information about the different types of relationship between genes, like autology, paralogy, and the gene tree. Otherwise, they do not provide information on transcript evolution, such as splicing autology and transcript phylogenes. So why do we need for a transcription database? So first, eukaryotic genes can produce several distinct alternative transcripts with different functions due to alternative splicing, which is a well-known mechanism that makes the transcript a refined level of functional homology relationships. Then, more and more data on transcripts are available in open source databases, and the information on their evolution can be useful to reconstruct accurate gene trees. So finally, different transcripts from one gene may have different functions and may be localized in different tissues. So more knowledge about transcripts can help to better understand the evolution of gene functions. So we propose TranscriptDB, a database and analysis tools developed in our lab. So TranscriptDB is a transcript database to study transcript conservation and evolution. It implements a new method to compute clusters of conserved transcripts in homologous genes. So what is the TranscriptDB content? So TranscriptDB contains initial gene and transcript data that were obtained from the ensemble resource, so the ensemble database. TranscriptDB provides different types of homology relations between transcripts, also transcript trees and gene trees. It also provides comparative genomics data display tools for data analysis and processing, and data export and a REST API to retrieve data programmatically. So TranscriptDB allows you to download data in FASTA or CSV format through its user-friendly web interface. So the collected and computed data are loaded into a custom PostgreSQL relational database. So the database provides relevant clusters of conserved transcripts and transcript phylogeny is computed thanks to a new similarity measure. So the TranscriptDB clustering algorithm is a graph clustering algorithm designed to identify conserved transcripts between homologous genes. So it takes at the input a set of homologous transcripts derived from homologous genes and gives at the output clusters of conserved transcripts. So the algorithm relies on two steps. So the pairwise transcript similarity measure, and also the second step is the graph-based clustering. So the first step, the transcript similarity measure, consists in first. So compute a multiple sequence alignment. So compute a multiple sequence alignment of transcript given at the input. So for that, we first align the subtranscript subsequences of genes. So after we remove all the untranslated regions, then we retrieve the alignment of transcript by mapping each transcript to its corresponding gene. So the multiple sequence alignment is decomposed into blocks, and a transcript can now be represented by a set of blocks. So a block is a maximum inclusive wide set of conserved columns. So then we compute the transcript similarity score for each pair T1 and T2 of transcripts. So the score is defined by this formula, where BI represents the intersection set of blocks and BU represents the union set of blocks. So there is a corrected version of the measure shown here that only consider differences at the transcript level by excluding those at the gene level, for instance. when we consider the transcript T21 and the transcript T11 derived from gene 1 and gene 2, we can see that the block 7 doesn't appear in gene 1, so the block doesn't appear also in the corrected version of the transcript similarity score. And alpha B represents the weight of the block, and the weight of the block can be unitary or associated to the length of a block. So the second step is the graph-based clustering that consists to first compute the symmetrical score matrix which represents all the pairwise transcript similarity score. Then we infer the recent paralogs and putative iso-orthologs. So two transcripts derived from the same gene are inferred as recent paralogs if the transcript similarity measure between them is higher than the transcript similarity measure with any other transcript. Two transcripts derived from different genes are inferred as putative iso-orthologs if they are reciprocal best hits. So then we construct a graph of homology based on the score of the matrix where the node represents the transcript and edges the homology. So we start with an empty graph where the recent paralogy edges are added. Then we added progressively the putative iso-orthologs edges if the property that the graph remains an orthology graph. At the end, the connected component represents the orthologs groups. So there is also an algorithm for the transcript phylogenes reconstruction developed in our lab. So the algorithm takes at the input the clusters of conserved transcripts from TranscriptDB clustering and the corresponding gene tree, and gives at the output a transcript tree. So the algorithm has two steps. So the first step is the transcript subtrees construction, and the second step is the transcript supertree reconstruction. So the first step construct the transcript subtrees in congruence with gene trees for each transcript cluster resulting from the TranscriptDB clustering. Then the second step is to construct a transcript supertree by combining all the subtrees based on the transcript similarity measure and the reconciliation with gene tree. So the optimization criteria is to minimize the number of creation in the resulting transcript tree. So the resulting supertree is used to infer homology relationships between transcripts. So here we have the overall view of the method proposed that combines the TranscriptDB clustering algorithm and also the transcript phylogenes reconstruction algorithm. So what type of data can you get from TranscriptDB? We can get transcript level resources like clusters of conserved transcripts, or transcript trees and stable ID mapping for transcript trees, the different types of homology relationships between genes, and the decomposition of the multiple sequence alignment into blocks and the conservation score associated. Also, we can get gene level resources like gene trees, gene splicing structure, so the gene model, and the set of exons, the types of homology relationship between genes, and the alignment of homologous genes. So the TranscriptDB web interface procures a quick access to transcript information. So here we have a transcript ID given by a user. We have automatically access to information about clusters of transcripts and information of corresponding gene, like the ID of gene, the stable ID of gene tree, the ID of transcript tree, the biotype, the strand of gene, the number of exons present in the transcripts. So the TranscriptDB provides also an interactive view of the exon entry structure of transcripts by highlighting the exons. The data can be downloaded by the user. The web interface gives a full perspective of the transcript sequences that gives a unique color to each exon to assure comprehensive exploration of the structure of the transcripts. So we have the description of the different types of homology relationship between transcripts. In addition to provide an interactive view of transcript tree, TranscriptDB provide a multi-scale graphical view to highlight the conserved transcript into the corresponding transcript tree. So then we can get gene-based resources in TranscriptDB. Indeed, we can retrieve gene tree and all information about homology between genes, like those mentioned for transcript. So we can see the exon intron composition of all transcripts produced by a given gene, and the TranscriptDB highlights the conserved exonic region in the red. So also the data can be exported. Here we have the transcript table that describe all the transcript derived from the gene given by the user. So all the resources are included into comprehensive and visualization tools that offer you a description of transcriptomic data. So to conclude, we presented a transcript-centric database that makes use of a new method based on splicing structure to first compute clusters of conserved transcript and then reconstruct transcript phylogenies. So the database provides tools for analysis and visualization of genomic and transcriptomic data. Database content is accessible through a REST API, and the tools is freely available through its web interface at www.transcriptdb.cobios.usherbrook.ca. So I would like to thank the Canada researcher, the University of Sherbrooke, and my supervisor and all the members of Cobios laboratory. It was a pleasure to present TranscriptDB at GLBIO 2023. Thank you.